So, well, first of all, thank you, Mario, for having me here. I wanted to say that you actually have a special responsibility in me moving from physics to biology, because for some reason journalists, Italian journalists in particular, think that anybody with a degree in physics can fix their TV. And so the only reason why Mario would normally call me was to get this
yes,
IT equipment.
A few decades ago, yes, he was very handy. I said, please, Andrea, do you mind? There is something not working in the cable.
So now
little did I know that Mark Zuckerberg would become interested in it, but here we are.
So, when I was at IBM in the first few years I was there, this is not just IBM, it's IBM Research, which is a very academic environment. We were doing research in artificial intelligence, and particularly machine vision. And what we found out is that all the algorithms that we were using to recognize objects in a scene were actually, as the first few sequences of both DNA and proteins were coming out, were actually really helpful in so determining what was the function of these genes or these proteins. And so we came up with a few algorithms that have been used quite extensively, one called Flash, one called Splash, that essentially helped characterize the biology of the entire human genome.
And one way or another, if I'm not mistaken, we're pioneering artificial intelligence,
yes.
also using artificial intelligence way back then. So
yeah, so it's a very interesting story, because artificial intelligence was very in the late 80s, and then became a four-letter word, and everybody thought that it was never going to succeed. And then finally, when
I think he means dead.
Yeah,
please don't think about other four-letter words. I'm sorry.
and now as an incredible comeback with these new large language models, and it's been quite exciting to see the progress.
Wonderful. Also, let me take a second to recognize with us Simona Vigna, who behind every man there is a wonderful lady, and Simona has been patient enough to go through what a scientist goes through. So thank you for having supported him. He would not be here without you. So let me address a second what I was saying about the fact that you are not a doctor and still you talk to doctors and you interact with, you're part of the medicine academy, national academy. Would it be possible in Italy that you would have this similar position? I mean, is interdisciplinary activity much deeper here than it is in other parts of the world?
Well, it's difficult to say, but certainly at the time when I came to the United States, it would have been almost impossible to do the kind of research that then led me to build the environments that I built. Mostly because the tremendous innovation that is there hasn't had really a receptor in industry itself. So here, when a university makes a foundational discovery, the IP that is generated by that discovery goes directly into the industry. And so in order to make the discovery nowadays, especially in biology, you need to bring together an extraordinarily vast range of disciplines to go from statistics to machine learning to and artificial intelligence to technologies that are used to now map genomes, etc. And then of course knowledge of molecular biology. So the only way to actually get it done these days is to create these very large interdisciplinary groups and research labs that can bring this thing to bear on the specific problem of interest. Think of it as putting a man on the moon. You don't do it just with one engineer that knows how to build a capsule. You need people that know how to rocket fuel works and people that know how to draw trajectories, etc. So it's complicated.
So is this one of the key, how can I say, feedback we should get from being here, that now medicine, it's a much complicated and larger environment, especially with the advancement in technology that has helped a great deal.
Yes,
for machines.
This goes straight into your second part of your question, which is, how does a physicist ends up running clinical trials, right? And the reality is that clinical trials now are increasingly dependent on sort of using predictive technologies that will tell you whether the patient will respond or not to a particular graph, whether there's an actual biomarker, and the physician themselves don't have the foundational expertise to be able to design those. So we're creating a new class of individual that we call physician scientists that essentially are bridging the gap between the basic science researchers. We're talking about Riccardo Dalla Fabra, for instance, or my lab, and the clinical labs. And so this really helps us participate directly into both the design and the analysis of the data that comes from clinical trials.
So you do your research, and by the way, I'd like to talk here at GEI also down the road, also a bit about philanthropy and the role of philanthropy. And and the fact is that you have been chosen by a philanthropic organization or by the philanthropic side of Mark Zuckerberg to run the New York program of his medical effort. First of all, let me ask you, so you met him directly? You had been interviewed by him? And
I met him several times.
Tell us, I mean, I don't know how many of us have, I haven't met Zuckerberg yet, but maybe you can tell us, how is it?
So first of all, people don't realize how young he is.
We understand that he's paying your salary, but anyway.
Well, he's not paying the Biohub is paying my salary.
Yeah.
He gave the money to make that happen. By the way, I'm sure you know that we raised about $260 million, and tonight we're trying to actually double that with your participation. So this is the entire goal of the union.
Yeah, no, exactly, but let's go into that.
So Mark is, people don't realize how young both Mark and Priscilla are. Mark is actually turning 40 in about a month. And so somebody who is basically one of the richest, if not the richest man on earth, at that age, has a different perspective on the kind of things that their philanthropy can do. And so when you talk to him, he's absolutely very approachable, very simple guy. Goes around in a t-shirt. Doesn't wear any kind of formal wear. You can talk to him about anything. And so we played guitar together. Everybody left the room.
Poor him.
Everybody left the room when I played.
Exactly.
No, I think they're extraordinary. And also, I want to say, I don't know many people that are willing to give 98% of what they own to philanthropy, right? So this is their commitment. Is just simply extraordinary. I mean, I think Warren Buffett and Bill Gates sort of started that trend, but I think he's been one of the people that have drunk the Kool-Aid and probably made the biggest commitment.
And so, but how did the process work? So how come, we would assume that if Mark Zuckerberg set something up like this and is looking for the president of his organization, there would be a great deal of competition. And how was the process? Did you go to a competition? Did they come to you? How did it work? So how does a philanthropic organization?
So Mark Zuckerberg and Priscilla Chan founded a Biohub in San Francisco about 6 years ago, and that was very successful. And so as a result of that, and that happened just because he knew the two guys that then became president, Steve Quake and Joe DeRisi. So there was no competition, but because it became so successful.
And that hub was doing what?
That hub initially was dedicated to using essentially a novel way of funding science to do the most exciting science possible, right? And then became more geared, and so his mission is sort of geared towards using immunology to study infectious diseases and autoimmune diseases. So as a result of that being successful, they decided to launch a competition, and there were 174 institutions that participated. We put together a proposal with Columbia, Rockefeller, and Yale, and that proposal was selected as semi-finalists. At the time, the dean of Columbia was just arrived, asked me to lead the Columbia side, and then we had co-lead from the other institutions. And so we moved from semi-finalists to finalists. Then you have to write a full proposal, and then once they decided they wanted to get funded, they had to decide who was going to be the president. So now they interviewed all sorts of people that our institution put forth. And I guess they decided that, I think this was largely based on the fact that I had experience sort of in building sort of organization like both for companies and departments, the center at IBM. So they wanted somebody who is not just a scientist but actually can get things done.
Yeah, you said you had the experience in building departments. So how did the passage happen to Columbia University? Again, I'm sorry, it's just to understand and to explain. Again, you were doing something totally different, and then you landed the medical department at Columbia. So it's a big step. How did that happen after IBM and after?
Well, so when I was at IBM, the one thing that I was missing and it's something that I realized you could not do computational biology without, was actually the experimental side of the lab. And IBM, because of building codes and a variety of other reasons, they were not willing to create labs for biology. So they would give us enormous amount of money to do computational research, but not experimental research. And so in 2000, I decided to leave, and I started, at the same time, I had spent what you call a mini sabbatical. So they allowed me to spend 2 days a week at Columbia, and that's when I met Ricardo Dalla Favera, and we started collaborating, and we have a lot of publications together. And so in 2000, I had a choice because Columbia had offered me a position, and at the same time, I wanted to try to build a company which was going to be the first company in pharmacogenomics called First Genetic Trust. And so I decided to go that way, and unfortunately, as you all know, then September 11 happened, and so the company went from being a discovery company to being essentially a service, like a CRO, clinical research organization, to a pharmaceutical company, which was not that interesting for me. So at that point, I called people at Columbia back and said, still have that position open? And said, yeah, come over. And this is one of the things that in Italy would never be possible, because basically it's like running a football team. The manager makes decision about who they want to hire or not, and there's no necessary due process. So it's
not a competition or grades.
It
or
wasn't a
points
competition.
or- yeah,
And so this is one thing which they recognize the kind of person they want, and if they find the right person, they bring them in.
So back to the foundation, the Zuckerberg Foundation. So then they decided to open here in New York, and they've chosen you. And being an Italian organization, we're very, very proud of that. But what then is the mission, and how different is the mission from the one of San Francisco? Can you describe the mission of the Zuckerberg Foundation and the so-called Zuckerberg?
The Biohub in New York.
Biohub in New York and the others they have, and the Zuckerberg project as a whole.
So in theory, the Biohubs, there's now essentially 4 of them, although one is called an institute, but it's essentially not the 4th Biohub. Is really a mission of taking on.
They're all one in San Francisco, one in New York?
2, one in San Francisco, one in Redwood City, one in Chicago, and one in New York.
Okay.
And I think they want to open new ones, potentially in Europe. So the mission of the individual Biohubs is defined by whoever made the proposal, right? But for some strange reason, essentially all the 3 main institutes, the 3 main Biohubs, ended up gravitating around immunology one way or another. Immunology is really probably the most exciting frontier of discovering in biological sciences right now. And the way we study that is the idea, we look at biological matter, at cells, as if they were programmable matter, computers, where you can actually change the code inside the cell and now make it do things that it was not able to do before. Right? And so some of the things that we've been able to do is to convince certain cells that create an immunosuppressive environment for the tumor, so the tumor is no longer recognized by the immune system, not making them able to go there anymore, and when you do that, this tumor is going to spontaneous remission. So this is an example of reprogramming a cell from what it normally does to something new.
I think you have to pick a little slower because we're getting technical, and it's extremely interesting. So can you tell us again? So
Sure.
You program the cell in a way that.
So you use genetics, right? So
Yeah.
you probably have all heard of CRISPRs, right? CRISPR is a new technology to silence or activate just specific genes, right? And so what our technology allows us to do is to find out what are the actual genes or proteins that are responsible for making the cell go from your bone marrow to the tumor. And then once they go to the tumor, they block the ability of the immune system to see that tumor. Okay? So if you now reprogram these cells, which are called regulatory T cells, you block the ability of the cells to go to the tumor by changing just one gene, silencing it. Now the tumor is going to spontaneous remission. Okay? So I'm just giving this as an example of what you can do, what we call cell-based therapy. So what we're trying to do is to really, so think about it this way, the immune system has been shaped by evolution, right? So evolution takes about 200,000 years to fix the genetics of a new life form into the population. So you have to think of what, and also evolution couldn't care less about what happens after you are in a reproductive age. So what happens after you're in a reproductive age 200,000 years ago has now been written in the immune system, right? So what was the reproductive age of 200,000 years ago? Probably 15 to 20 years old. So anything that happens to us, Parkinson, Alzheimer's, tumors, fibrosis, any kind of aging-related malady like senescence of the cells, it's not something that the immune system has been trained to recognize and to fight. So what we're trying to do is to become the new teachers, the new evolution in a box that will teach our own immune system new tricks so that it can actually take care of the things that it has not been trained to see. Does that make sense?
Makes sense, thank you, very clear. And so, on an overall project, so what are the objectives of the Zuckerberg project?
Yeah, so there are
What does Mark
four
Zuckerberg
programs.
have in mind?
Well, Mark Zuckerberg essentially has a committee of scientists that determines the quality of the science that you propose. So he's not involved. One of the things he tries to do is, and I think is a good thing, is to inject more and more of artificial intelligent approaches, which actually was part of our proposal, because studying the immune system actually is very, very potentially, AI is very potentially helpful to that. But it's not directing you to do certain things or other things, right? So it's done by the committee of scientists. And so once the Biohub is started, what the Biohub does is up to you. There's no intervention. There's complete freedom of operation. Of course, if the results are not good, then they can change. They can find another president. But at least until things are not going well, they're stuck with me. Okay.
But the long-term objective, I seem to remember that
Right.
you told me, is that Zuckerberg wants to defeat every kind of illness
By the end of the century.
by the end of the century. Now, that is a rather aggressive statement, which of course we welcome. I will not welcome it because the end of the century is beyond any reach. I was hoping that you would say in the next 5 years, so that would but so what does that mean? I mean, because new illnesses will come. I mean, we've seen COVID.
So you have to think about the acceleration that molecular-based medicine has had over the last 100 years, right? So the way you would be able to treat one disease at a time 100 years ago was largely based on serendipitous findings. You would, put a drug in a person and they would respond without any kind of knowledge about why they would respond. And for instance, I assume, as I'm aging as well, prostate cancer, the use of androgen deprivation therapy, it's completely serendipitous. There was no biology behind it, right? It was now we understand why it works, but the drugs were discovered, completely randomly. So what is happening is now that you have more sophisticated molecular biology, AI, computational approaches, is that you're starting to learn the mechanisms that drive different diseases. And so what you can do, you can literally accelerate the pace at which diseases will be treated. So when we say by the end of the century, it means that right now what we're trying to do over the next 10 years is to create the foundational infrastructure that will allow us to take care of maybe 2 or 3 diseases. And we are clearly focusing on some cancers and Parkinson, Alzheimer's. And then based on that infrastructure, you now can accelerate the pace of treating virtually all other diseases, because now you have both the technology, the ability to go and look at individual cells in a body, tackle their genetics, understand why a particular disease starts. So it's not mission impossible. It's just very ambitious, of course.
So, and how much did he fund your particular research project and the overall?
So the gift was a $250 million plus an additional $10 million to fund investigators that are in a particular class called HHMI. So $260 million in total. And so this is for 10 years. So it's about $26 million a year. And that basically is dedicated to creating an infrastructure that is both what we call an intramural, meaning that there's going to be an actual building with a fully dedicated floor, and between 60 and 65 people working there. And then what we call an extramural program, which will actually fund at the rate of about $1 million each, for 5 years, 24 investigators, 20 to 24 at the parent institutions on Rockefeller, Yale, and Columbia, plus potentially other institutions in the New York area.
So what happens if a cure is developed? Or let's say that you find a cure. You are there also in conjunction with Columbia, Yale, and so will that be commercialized? Will that be given to a pharmaceutical company? Will the foundation get money for that to fund more research?
Yeah, that's a great question. So It works very similarly to the way it works in academia. So we don't necessarily do clinical work directly. So for instance, I made certain discoveries that have been patented, and then those patents have been licensed to different organizations. And so the same thing will happen. And the reason we actually are patenting, so the Chan Zuckerberg initiative and the Chan Zuckerberg Biohubs are dedicated to this concept of open science. That is, we will publish our data the moment it's generated. We won't even wait for the papers to be written, which is a very novel idea because scientists just hold on to their data until their results have been published. So we want to do open science and make everything available to everyone. But the one thing that we want to make sure of is that somebody will be able to then transform the discoveries into actual implementation that will benefit the public. How do you do that? In order to convince a company to actually invest hundreds of millions of dollars to develop a new drug, you need to give them some kind of protection, otherwise they will not do it. And so the IP is purely to protect the discovery so that they can be translated. And any funds that will come from that will go back both to the universities and to the Biohub, and it will be used to fund additional research. So there's no commercial angle in anything.
Okay, I'm going to move into a couple of questions, but please I will open it up to you if you have any questions that you want to ask Andrea. So, Andrea, so more or less there is $1 billion between the four organizations, it's equally split. So you mentioned 3 illnesses that are on the forehead of vast majority of the people: cancer, Alzheimer, and Parkinson. So what can you tell us about that? You said, maybe in 10 years we will do something. What kind of research are you doing, and what kind of immune, are you pursuing the immune angle, and what can you tell us about it? Maybe let's start with cancer.
So, so the idea here is to build the equivalent of a molecular endoscope. So when you're trying to do a complex operation, if you have to open up the patient, it's very invasive, right? But if you use an endoscope, it's actually less invasive. And what we're trying to do is to build a molecular endoscope so we don't even have to do anything invasive at all. So what we're going to do is reprogram the immune cells in our body so that they can go to specific organs, sense whether there's anything wrong in that organ, and then report back. And to do that, there's a very large range of technologies that need to be brought to bear, ranging from the fact that we literally have to create special cells that can go only to the pancreas or only to the brain or only to other organ sites. The fact that we have...
So you said create special cells, meaning
correct.
These cells are not, they are
the cells engineered. Are cells from, they're engineered. That's why it's called cell therapy. They're basically cells that are harvested from the blood of a patient. And we can do either, more complicated, it's something called an allograft, meaning that you modify the cells of a patient and then you inject them into that patient. Why? Because otherwise the immune system, if you inject them in another patient, the immune system of the other patient will kill them immediately, rejecting this. But we are also trying to create cells that are, we call them universal. So if you remove a certain set of proteins called HLA, and if you actually activate some other proteins called a don't eat me signal, you can actually create cells that are invisible to the immune system, and well, not forever, but for a little bit longer. And so you can create kind of this universal library of cells that are specialized to go to the pancreas, to the brain, etc. So once you are there, now you have to sense the specific signals that tell you that there is a tumor or that there's an incipient Parkinson, etc. And so for that, we have to further engineer the cells to instrument them with, we call it, so the cell has a membrane. You need to stick something in the membrane that can sense the signal that the disease is emitting. And then you have to build a memory device inside the cells that will record that and bring it back to the doctor, right? So it's a multi-step process, extremely complex, but the most amazing things is that all the little ingredients that are required for implementing that vision are actually already available. It's a matter of really bringing them together and functionalizing them. So I think this is what makes a proposal that would otherwise sound like science fiction to be actually probably feasible.
Let's open to the public if we have questions. Galeazzo and Anthony. Yes.
Especially in the US, chronic disease is enormously a function of social determinants of health and population health and prevention. So to which extent your programs will look into social determinants and population health, which is more, why do we have so many chronic diseases like cancer? You know, how many pesticides do we put in our food? We clearly determine the amount of cancers we generate before we start curing them.
There's no place like New York in terms of its heterogeneity of the population. We speak, I think, 156 languages in the city. And so this is absolutely first and foremost. You know, we will not create our own clinical study in the Biohub, but because of the liaison to the parent institutions, we will have access to populations, cohorts of patients with cancer, et cetera, that actually reflect the true heterogeneity of the population. So this is a critical element.
Final analysis, what you're trying to do is to prolong life. Have you thought of the economic and social consequences of that?
It's a great question. So right now we're trying to prolong life of people like children with rhabdoid disease that would actually become very productive members of our society. So I think that the goal of prolonging, you know, the normal lifespan of a human being is well far off. So we're trying to do things that intervene on things that go awry during our life, as opposed to things that have to do with our normal lifespan. There are economic consequences even in that, but I think one of the biggest burdens that we have right now in this country is actually the treatment of chronic illnesses. That has an incredible draw on the economy, especially in the United States, where, you know, essentially health treatment is much more expensive than in other places. So I think that I'm hoping that the two things will balance out, or in fact that actually prolonging life with less dependency on treatment of chronic illnesses will actually help us even win a little bit.
Sure, you have heard and you know a lot about in vivo therapy. Okay, and my question is, can you elaborate on that? Because a lot of people are confused about this in vivo therapy and also the risks involved, you know, with this type of therapy. Also, that is very interesting what you said. Unfortunately, life is true, it's a longer life. But unfortunately, the longer span is not between 20 and 30, it's between 80 and 90. So people have the, 100, 110, have the right to live those years, those longer years, the best condition, you know, quality of life is, exactly. So thank you so much.
I think when you say in vivo, you mean you put a drug in a patient. We will not do clinical research in the Biohub, but we will collaborate, as I said, with both pharmas, you know, biotechs, and the universities to actually develop the foundational molecular biology concepts that will be discovered at the Biohub into cohorts and to tackle disease.
First of all, congratulations. Question number one, you mentioned when it comes to Italy, the limits or the blockage in terms of career. Could you give us an assessment of what the state of the research is in these fields in Italy, number one? Question number two, and again the journalist here. I wonder to what extent what you're doing and what others are doing in this field, even just in the US, to what extent is coordinated or is not coordinated? In other words, now we heard what you're doing, the whole project, the investment, and Zuckerberg and all that, but is there any kind of coordination with other labs, with other universities, or everybody's doing his own way in this?
I think that one of the biggest problems that is in some way depleting the ability of Italy to be at the cutting edge is the fact that the science pipeline is actually quite solid. We also have some terrific physicians, you know, so some of the clinical trials conducted in Italy are among the more interesting in the world, but we lack the translation into industry. And so, you know, I don't remember who I was talking to, but the number of patents that are generated by Italy is very large. The number of patents that are exploited by Italian companies is extremely small. Right? And so I think, and the problem is with that is that the flow of money that is generated by the licensing of the patents flows back into the science. And so to some extent, I think breaking that barrier is going to be very important. The other things that I've noticed is that in Italy, doing science is considered to be a privilege. It's not considered to be a job. So basically they say, oh, we put you in a room full of toys and we even pay you for it, so what do you want? Like, leave us alone. And I think this is really the attitude of the government, and the investment have been mostly in other areas, from fashion to tourism, etc. So I think that when you go to in a United States institution, there is a competition. So just give an example. To retain one of the key members of my department, we had to raise his salary to $350,000. Now, who in Italy is paid $350,000 a year to work in a university? No one, right? It doesn't exist. So the ability to put together teams that are more akin to putting the best soccer team together and pay whatever the competition forces you to pay, I think is going to be an absolutely critical element of maturing the science. Of course, you can't do that if you don't have the revenues coming from industry, et cetera. So it's all connected together. In terms of the second questions, remind me for a second what coordination. Yeah, so the coordination here is remarkable. For instance, let me give an example. The other side of Chan Zuckerberg is called CZI, Chan Zuckerberg Initiative. So they have been the ones that have created this universal effort that supports groups in Africa, Asia, Europe, and Asia Pacific, etc., to actually put together an atlas of single cells from all the normal organs in all these different populations. So that's called the Human Cell Atlas. This is something that requires coordination of literally hundreds of labs. They've given probably more than $500 million to $1 billion just for that. No, forget the Biohub. So this is an example of the kind of coordination that you can do when you put your money behind an idea and you drive it. Now, this is very different. I think I want to get to that because I think it's an important point. The funding model that the Biohub is having is fundamentally different to what you use in a university and also for CZI. The idea is that in a university, each individual will write grants, and the grants can be funded or not funded, and many times you get amazing grants that are not funded for completely serendipitous reasons. And this forces individual to actually work as in a monadic form. It's like my lab is against your lab, right? In the context of the Biohub, all the work is funded up front. You don't have to write grants. You don't have to do anything. Of course, there is a review process. If you don't do well, we'll kick you out. But the idea is that you should only worry about the science. You should worry about the fact that you're proposed to do something transformational, and that you now go and sit at your lab bench or computer and you go do it, right? The amount of money, the amount of time and money, because you have to generate preliminary results, that you spend in getting funded is extraordinary.
In the universities,
yeah.
So you become a fundraiser, not just a scientist.
You become essentially like an entrepreneur. It's like labs in university in the United States are mini companies, and they're trying to get money from all possible different sources. Every time they're struggling, 1 year goes bad, you have to fire half of your lab. Now just recruiting it back is a disaster. So there are all these problems. Here we will have continuity. We'll have a 10-year, and San Francisco has already been extended for another 5. So they want us to work until the end of the century. So I think unless it doesn't work, which hopefully will not be the case, this really gives us a model that is very different.
So I'm kind of curious, what organizations do you like working with the most, and which do you find more competitive? If you talk of the top 10 organizations in the world.
Sanger Center in Europe and UK are extraordinarily collaborative, and so they create enormous data sets. They also benefit from having a fully integrated health system. I mean, one of the big problems we have in the United States is that the patient at Columbia and the patient at NYU and the patient at Mount Sinai are not shareable. Each one has its own court and all. In UK, if you're in the system, you're in the system. Every single person is in the same system. And so there are things that they can do, like sequencing 500,000 human genomes across all possible disease areas, normal behavior, etc. Is just unprecedented. So they're great at sharing that information. And in fact, there have been now recent initiative with the CRUK, Cancer Research UK, and NCI to fund what is called the big challenges, the really major challenges in cancer. Other organizations are more sort of self-inbred, and I don't want to make names because otherwise I'm going to make enemies. And we certainly want to correct the behavior, and we hope we will be able to. But they're basically feeling that by collaborating with you, they're essentially reducing their own visibility. And so that is a problem, and I think something we have to overcome. Certainly our mission is to be collaborative with everybody. In fact, to just share our data so that anybody can use it.
Okay, then I see there are no others. I have a last question for you, Andrea, which is more
I hope
short
it's
term.
not about fixing your TV again.
It's now digital, you wouldn't be able to do it. It's too complicated, even for you. But no, it's related to the short term rather than the very long term. And you were saying at a certain point that one of the issues in attacking one cancer is to identify the movement or how to reach it. And so, what can you tell us for the very short term so that we say goodbye with some sense of hope?
So, well, I think an element that is going to be unique for our Biohub compared even to the other three is that we're going to be the one using this very, very intersecting connection between computational sciences and biology. And that allows you to create models that are predictive. So you can actually go and say, oh, this patient should be treated with this drug. And now we've tested that just to show you, to give you an example, in 2 cancers that are extremely aggressive, metastatic neuroendocrine tumors of the gut and rhabdoid and Wilms tumors, malignant metastatic in children. We've had clinical trials and that have completed, another one in metastatic breast cancer, completed with 100% success. So, and this is the, for coming out as a physicist.
I'm sorry, I didn't understand. How did you do it? You treated it
So we predicted that based on the
Certain
computational model of how the cell works, which drug should be used to treat this particular disease in this particular
So
patient.
super personalized.
Right, so it's personal. Well, it's personalized enough that you can now split a cohort in say 30%, 30%, 40%, and say this 30% should be treated with one drug, this 30% with a different drug, this 40% with a different drug. And these results have been quite remarkable. Like for instance, we said, if you have aberrant activity of these particular proteins that you can only find computationally, you will respond to this drug. And 100% of the patient that had that activity responded, 100% of the one didn't have that activity did not respond. So that's an example. So I think this is really changing the speed at which we can foster clinical trials. To some extent is what my companies also do with Darwin Health. And we think that is going to be absolutely instrumental in changing the way we think about reprogramming the immune system to go and discover early signs. So I don't know if you know, but for instance, the medicines, the drugs that have been developed for Alzheimer's and Parkinson are actually reasonably effective when they're used very early, right? Not effective at all when the disease has progressed. The problem is that it's almost impossible to detect the disease early. So if you can actually use the immune cells to actually go and discover.
You were saying that Alzheimer before going in the brain is.
I'm sorry, what were you saying?
That Alzheimer before finding it in.
No, what was your previous question?
It's down in the stomach or
No,
in the
it's
belly?
not Parkinson. The first signs of Parkinson are actually in the neurons in the gut.
Yeah.
And so, it's a little problematic to send things behind in the brain because there's something called the blood-brain barrier. It's something that protects the brain so that things don't go in there. There's a lot of immune cells that go in the brain, but instrument them to go there, it's probably harder. So if you can actually instrument the cells to go to the gut, it's a lot easier to detect early signs of Parkinson. And that's also where the first signs are detectable.
Well, thank you. The most politically incorrect scientist we have